Don't worry if you're wrong. It's not like you're going to lose marks for it. It is true. Why is it true? Well, let's think about it. Can you personally absorb solar energy and make ATP from it? Are you a plant? Do you do photosynthesis? No, you can get a tan, but that is not going to make your muscles contract. And so I want you to see the link here that for solar energy to end up helping a muscle contract in your arm, I had to get ATP. Chemiosmosis is the way ATP is made. Now, there's a couple of spots it would happen. First, it happens in the plant during photosynthesis. Second, it happens during cell respiration. So absolutely, chemiosmosis must occur because it's ATP that's going to make your muscle contract. So that's why 1 is true. How about number 2? Does anyone want to tell me if that's true or false? The overall reactants for the photosynthesis reaction include CO2 gas and H2O liquid. It's true. That's what they are. They are the things that start the photosynthesis reaction. I could also have written they are the products for the cellular respiration reaction. Now, number 3 is false. It's true that I produce them at some point during photosynthesis, but I use them up. So when I say the overall products for photosynthesis, what I'm referring to is not those 2 substances that get made and then used, but I'm referring to oxygen and glucose. So those are the overall products for photosynthesis. They're also the reactants for cellular respiration. Number 4, is that true? ATP provides energy by releasing 3 phosphate groups. It has 3 phosphate groups. That part is true. Does it release all 3 of them when it's giving energy? I see some no's. This is false. It provides energy by releasing 1. ATP turns into ADP. That's where we get energy, from the bond between the 2nd and the 3rd phosphate. Number 5 says NADPH has reducing power, while NADP positive is an oxidized molecule. That is true. NADPH has an H. If it has an H, it is reduced. It has reducing power. If it's missing the H, then it's oxidized. Now 6 is asking you about the diagram. These 2 are opposite. On the picture, A is not the thylakoid. A is the stroma. And on the picture, B is not the stroma. It is the thylakoid. On the picture over here, C is the matrix and D is the cristae. But the first part is mixed up, so that would be false. You can see that I specifically put a picture of a chloroplast and a mitochondria. That would be something that I would probably use on a quest, is a picture. Then I ask the function of part A, which we just decided was the stroma, is to reduce carbon dioxide. This is true. Carbon dioxide happens or is used in the Calvin-Benson cycle. That's also called the light-independent reaction, or the second part. And the second part happens in the stroma. So that one is true. Number 8 is false. The reduction of NADP positive doesn't happen during the Calvin cycle. It happens during photolysis, or the light reaction, the first part. Does anyone have any questions so far that they want to ask? Number 9 is true. I make ATP in photolysis so that I can use it in the Calvin-Benson cycle. 10 is false, and it's the word absorbs that makes it false. Plants do not look green because the plant is absorbing green. Plants look green because the leaf is reflecting green. Number 11 says the light-independent reaction occurs during the day and night in all eukaryotic organisms. That one? Oh, I was being mean there. If I was asking you about cell respiration, that would be true. But I'm asking you about photosynthesis. That's false. All eukaryotic organisms do not do photosynthesis. Only ones that are autotrophic, like a plant or an autotrophic bacteria, would do that. Now, number 12, we'll say is essentially true. The way it's worded isn't the best way. It's really the movement of hydrogen ions that is helping produce ATP, but that's the association that I want you to make. Hydrogen ions move, that's how I get ATP made. There's more to the story, right? There's ATP synthase, that structure that's in a membrane, and the hydrogen ions have to move through there down their concentration gradient, but that's the link. Hydrogen ions are your chemical substance. They are moving chemiosmosis. Number 13 is true. To replace lost electrons, photosystem 2 decomposed a molecule of water. That's true. So photosystem 2, remember, is actually the first one, where light hits, electrons get excited, and they start moving. Photosystem 2 wants to put electrons back, so it breaks apart a molecule of water. Oxygen is produced in the thylakoid membranes while glucose is produced in the stroma. That's true. Oxygen comes from photolysis. Glucose comes from the Calvin-Benson cycle, and that's where they both happen. 15 is false. It's the other way around, like I just mentioned. Photosystem 2 comes first, then photosystem 1. If I was to trace the path of electrons, they start at photosystem 2, they go through some carriers, and we didn't name the carriers. And while they're moving, they help hydrogen ions keep their gradient. They end up at photosystem 1, and then they end up reducing NADP positive. That's the path that the electrons are taking. Number 16 says to produce NADPH, electrons are required to oxidize NADP. It's false because of the word oxidize. If I want NADPH to be produced, I have to reduce NADP positive. Electrons are required, but it's the word oxidize. If I had said reduce there, that would be true. Number 17 says, in the light-independent reaction, carbon dioxide is incorporated with RuBp to form molecules with 3 atoms of carbon first, and later molecules with 6 atoms of carbon. It's essentially what it's happening. I left out some of the details, but when one Calvin-Benson cycle happens, I get a 3-carbon molecule, and when the cycle happens twice, I get a second one. There's my 6-carbon glucose molecule. So that was a very generic description there of what was happening, but that's basically what's happening. It has to happen twice, because one time only makes a 3-carbon molecule. It's the second time around that I get the 6-carbon molecule, which is glucose. 18 is true. An increase in temperature could increase the rate of photosynthesis. And I'll just add, but only to a maximum point. Remember, once it gets too hot, enzymes are denatured, and the rate of photosynthesis drops right off. I'm going to point out thing number 1. Thing number 1 asks about energy getting from the sun all the way to a muscle. On the very first lesson about photosynthesis, we talked about a cycle and how the sun comes in and it goes to a chloroplast, and that makes oxygen and glucose, and it goes to a mitochondria, and that makes ATP, and ATP has functions. It was one of the textbook questions that you had to answer the other day. Trace the path of energy from the sun to a contracting muscle in a mountain lion, I think was the example there. I like that question. I like it a lot. A lot. Enough to maybe say give several marks as a written question about this topic. The other thing that I recognize is that each step of photosynthesis has a whole bunch of little parts in it. And so I regularly like to take one of the 2 parts and make it a written question so that you can tell me what you know about it. Maybe you forget one of the details, but you remember some of the other ones. Photosystem 1 and 2, so photolysis. I'll just circle photolysis. Photolysis, I think, is the one that's a bit better for explaining. In photolysis, I left out the names of molecules, but not really the processes that were happening. In the Calvin-Benson cycle, we really simplified. If I was going to ask a written question, I would ask about the one that you know more about, photolysis. All right, so we are on the third little section of this unit. Realistically, I guess photosynthesis and cell respiration go together, but I'm calling it the third section because we're doing a quest in between it. There are only a couple of lessons in this third section, so it's not particularly long. We're going to focus on cellular respiration in this section. And so we'll start the same way we started with photosynthesis by talking about how we get started. What's the overall idea? So we'll start with lesson 10. Lesson 10 is where we're really going to focus on glucose. Where does glucose go? How does it get to the mitochondria? And what happens to it? And then we'll focus on oxygen in some of the later lessons. But we're really going to focus on glucose today, which is why I called this one Glucose's Destiny. So we will start by just filling in what I get or what I use during cellular respiration. Remember that this side is called reactants, and this side is called products. So the reactants for photosynthesis and cell respiration we already talked about, but so you remember, I have 1 glucose. So I'll write the word and the formula so you can recognize. Glucose is C6H12O6. So I have 1 glucose, and I have 6 oxygen gas molecules. As my products, I have the same things I would start with for photosynthesis. My 6 molecules of water and my 6 molecules of CO2. So we have water and we have carbon dioxide. Now, aside from those substances, which are the main reactants and products, I like to specify, because we mentioned it at the beginning, that there's some waste heat that's being produced. When your body warms up, it is because of cellular respiration. That's what's making you warmer, is the waste heat from a metabolic process like this. The other substance that we're going to focus on is ATP, because unlike photosynthesis, the ATP that I make during cellular respiration doesn't get used up right away. It gets transported off to your cells so that they can perform functions with it. The other thing that I want to remind you of, even though we've already mentioned this, is of the parts of the mitochondria. You have the matrix inside. You have the internal membrane, which is the cristae. And don't forget that outside line is just the membrane of the mitochondria. So those are the main things that are reacting and being produced. And the 2 parts of the mitochondria that we would focus on, because we're going to talk about which processes happen there. Does anyone have anything they want to ask about the overall idea of cellular respiration? Okay. So even though I'm not specifically going to ask you about all the details of cellular respiration, this overview really we talked about already. And some of the stuff that's on here I could ask you on that quest that we're doing tomorrow because we already did it. We're just repeating ourselves here. So what we'll talk about first is glucose. Like I said, we have talked about where glucose goes. And we're talking about glucose metabolism. So that means the reactions that involve glucose, or the reactions that are necessary to get glucose available so that your body can do cellular respiration. So we start with our poly and disaccharides, and I'll just remind you, poly means many, di means two. So we start with our big sugar or carbohydrate molecules. Are broken down eventually by enzymes into monosaccharides. Mono means one. So regardless of what format you eat your carbohydrates in, eventually they're all going to get broken down into monosaccharides. Then your blood comes along and delivers them to your liver. We will talk at length about your blood and your liver when we're talking about circulation a little bit later on. Does everybody know where their liver is? Like if you looked at yourself, where is your liver? Okay, well let's make sure you know. Your liver is kind of right under where your rib cage is, on the top, on the right. That's your liver. Your liver is quite enormous, like it's a huge organ. You'll see when we do a dissection how big the liver is compared to other organs. And most people don't have a really great idea of what their liver does, and so the answer, everything. Your liver is part of every system that we're going to talk about. Your liver is obviously part of digestion because it's helping with carbohydrate transfers. Your liver is part of circulation. Your liver also helps detoxify your blood and break down blood cells. Your liver is where bile is produced. Your liver produces the substances that come out in your urine. Like your liver is everywhere. And a lot of people don't realize quite how important your liver is. You know, you think about the big ones, heart, brain, and you don't realize quite how essential your liver is. Your liver takes fructose and galactose, which are both monosaccharides, and converts them to glucose. There's some steps I would never ask you, but there's a little reaction that happens where we just rearrange a few molecules and turn the fructose or the galactose into glucose. So eventually, whatever carbohydrate you consume will be glucose in the long run. Sometimes it takes a few steps, but that's what it'll be. And then your cells absorb glucose from your blood, specifically tiny little blood vessels called capillaries. When you have a bruise, your capillaries are broken. That's what you can see. A bruise is a bunch of little broken blood vessels, and it's capillaries. They're really thin, and that's why they're good at transferring things into your cells. So you'll see the picture on the side here is talking about intake, is talking about storage. So I'll just mention this. We already know that glycogen is a polysaccharide that animals use to store carbohydrates. Since we're not really getting at storage here, we're getting at usage. I didn't put it in the notes there. But eventually, my big carbohydrates and my disaccharides become monosaccharides. And then finally, it becomes glucose, and it is distributed and used. Does anyone have questions about how glucose would get to your cells? Some of this will come back for sure when we talk about digestion, and again when we talk about circulation. So glucose goes to a number of places. So once your body has processed it and you have glucose available, there are 4 basic places that glucose can go. One of them is glycogen, like we just saw. Glycogen is how we could store it. So if your body recognizes, hey, I have more glucose than I require at this particular instant, instead of leaving it in your blood to circulate, it will get stored in the form of glycogen. Glycogen is what you have in your muscles, and glycogen is easily accessible. So glycogen is what your muscles have in reserve for the time when all of a sudden you start sprinting because a coyote is chasing you through the park by your house in Cimarron. This happened to me. This is a true story. I'm walking through the park, and I'm like, wow, that's a really big dog. Holy cow, that is not a dog. And I had my little wiener dog with me, and my dog got freaked out, and then the coyote saw us and started like running towards us, and I was like, uh-oh, I better get out of here fast. So I was using glycogen for sure. For sure. Fat is the other storage place. Fat is stored in something called adipose tissue. And this prefix here, that's what you would see when we're talking about fat. A fat cell is called an adipocyte, but that's your fat tissue. Fat has a really, really, really slow rate of metabolism. So it's less easy to get the glucose energy from fat. It's not impossible, it's just more difficult. And if your body has the choice, it will pick glycogen first, because it's easier, and wait till that is completely depleted before it starts using the energy that's stored in fat. One of the reasons that fat is so hard to get rid of once you have it. So these two are for storage. One of the things that we'll talk about a bunch during this whole course is the idea of homeostasis. So I'm going to remind you what this word means. Constant state. Your body has an optimum level of glucose that should be in your blood. If there's too much, your body doesn't leave glucose in your blood, it stores it. The way that this is regulated is typically insulin and glucagon. Most of you would be familiar with insulin. People who are diabetic often have to take insulin shots to regulate this. But glucose goes into storage because your body is trying to keep a constant state. There's too much in the blood, so some of it has to go into storage. Now the next one is excretion, and let me say that this is rare. There are very few circumstances in which you would be excreting, which means that your urine would contain glucose. A diabetic would be my example of a person who would have glucose in their urine. Your urine is a waste product. Usually, your body only puts things that it wants to get rid of in your urine. Your body would never voluntarily choose to get rid of glucose because it's the fuel that every cell requires to survive. And so the only time that we would find glucose in urine realistically is if the person's blood sugar was so incredibly high that there was just nowhere else to put all of this glucose. So you'll see when we talk about disorders in each of the sections on human physiology, diabetes will be the one that we'll discuss quite a bit because it has to do with not only your digestive system but also your excretory system and the production of urine. But generally, a test for sugar in the urine is a good test to see if someone is diabetic or pre-diabetic. The last fate of glucose is where I make ATP and some waste heat. This means that cellular respiration is actually happening. So there are lots of things that can happen to glucose besides performing cellular respiration. Now the heat is often called waste heat, which is kind of a bad term because sometimes the heat is actually very useful. We'll see in our next section on muscles that one of the ways that your body performs homeostasis is shivering. Shivering is when your muscles contract, so they do cellular respiration, and they produce waste heat on purpose to warm you up. So even though we're calling it waste heat, sometimes it's not bad. Sometimes it's actually very useful to have the heat that's produced from cellular respiration. ATP, I'm just going to remind you, in lesson 6 we went over the functions of ATP, so I'm not going to repeat these in the notes again, but they are things like muscle contraction, metabolism, synthesis of macromolecules, movement of cells that have a flagella or cilia. Those are all things that ATP can do as its function.